Welcome to Chapter 15, Genes and Proteins. We'll just be wrapping up genetics in the next couple of weeks in preparation for your final exam. So the central dogma of genetics. We're talking about the flow of genetic information from DNA to mRNA. Essentially, the creation of RNA from DNA to make proteins. Genes will specify the sequence of mRNA, which specifies the sequence of amino acids to make proteins. The translation of proteins is collinear and systematic, meaning that specific nucleotide bases pertain to specific amino acids. The genetic code is nearly universal. Each amino acid is equivalent to a triplet codon, and a codon is 3 nucleotide bases that are specific to a specific amino acid. The genetic code is degenerate, meaning that amino acids can be encoded by more than one triplet, and this was proven by Crick and Premrna. This reduces negative impact of mutations, as certain mutations may actually lead to just a different amino acid. Nonsense codons are stop codons, and they stop the process of transcription. So you see here on this chart that different triplets can code for the same amino acid, and these are just abbreviations of amino acids. Recall that in prokaryotes, they don't have a membrane-bound nucleus. In fact, instead they have a nucleoid region, since they don't have a true nucleus. They also have abundant plasmids, which are circular pieces of DNA that they can trade with one another to trade adaptations. In prokaryotic transcription, this requires the double helix of DNA to partially unwind. An area in which it's going to do this is called the transcription bubble. Transcription always proceeds from the same DNA strand for each gene. This will be called the template strand. Non-template strands are the mRNA product that is complementary to the template strand and is almost identical, except it's RNA rather than DNA. There is a difference in nucleotide base pairing from DNA to RNA, as DNA has base pairing from adenine to thymine, A to T, and from cytosine to guanine, C to G. In RNA, we have complementary base pairing from adenine to uracil instead of thymine, A to U, but we still have that cytosine to guanine, C to G. Upstream nucleotides are any nucleotides before the initiation site in transcription, and downstream nucleotides then would be the opposite. Any nucleotides after the initiation site. And then the initiation site is just where transcription is going to begin. So let's talk about the initiation of transcription in prokaryotes. Transcription, translation, and mRNA degradation can all happen simultaneously because there is no membrane in that nucleoid region to prevent these things from occurring simultaneously. They just can all occur at the same time. RNA polymerase, the enzyme, will transcribe all genes. It's very straightforward in prokaryotes. There's just one enzyme that runs all of this. There's a promoter that will bind and initiate transcription, and it will exist upstream from genes that are regulated. DNA will be continuously unwound ahead of the core enzyme, that RNA polymerase, and rewound behind it. Base pairing between DNA and RNA is not stable enough to maintain stability of mRNA synthesis components. So RNA polymerase, that enzyme that's our core enzyme, acts as a stable linker between the DNA template and RNA. This ensures that no interruptions occur during the process, which could cause mutations down the road. RNA polymerase must be instructed to detach from the DNA template when this is all over, and the creation of the stable hairpin, which is just mRNA folded in on itself, will help this to cease. This will cause the polymerase to stall as it begins to transcribe any A to T rich nucleotides. In eukaryotes, this process is a little bit different. There's 3 different RNA polymerases, RNA polymerase 1 through 3. The first one is located in the nucleolus and helps to make rRNA. RNA polymerase 2 is located in the nucleus and synthesizes pre-mRNAs. And RNase polymerase 3 is located in the nucleus as well, and it makes rRNA and tRNA. The eukaryotic promoters are a bit larger and more intricate than the prokaryotic promoters. One of the big ones is called the TATA box. The TATA because it's composed of thymine and adenine. There's also regulation of pre-mRNA synthesis via enhancers and silencers. There's a greater metabolic investment with eukaryotic transcription. So cells have to the cell prescribes exactly the pre-mRNAs necessary for this process to occur, so that it's not expending energy that it doesn't need to. Elongation is essentially the same as prokaryotes, but the DNA template is more complex, as can be expected. Pre-mRNA will go through an extensive process before it's translated into proteins because introns have to be removed or spliced from the exons to make translatable mRNA. If you don't take out those introns, it can be devastating to the organism and cause a variety of different diseases. tRNA and rRNA, transfer RNA and ribosomal RNA, are not translated, but they do have roles in protein synthesis. Mature rRNA makes up about 50% of each ribosome, and tRNA folds to position amino acids at one end and anticodons on the other. And the anticodon is the triplet in tRNA that interacts with the mRNA codon through complementary base pairing. So protein synthesis as a whole, which would be this whole process of transcription and translation altogether, consumes more of a cell's energy than any metabolic process. Amino acids will be strung together by covalent or peptide bonds to make proteins. And the nucleolus will create ribosomes, which are composed of rRNA, in eukaryotes. Ribosomes are also present in prokaryotes, but they're not created by the nucleolus. The small subunit will bind the mRNA template, and the large subunit binds the tRNA. So although the ribosomes are not in the nucleus, they are composed of and creating and binding to a lot of the components that help to make proteins.